Trabalhamos diariamente para criar soluções e equipamentos para a comunidade científica, contribuindo com o avanço da pesquisa biomédica latino-americana. Nossa missão é maior do que somente fornecer equipamentos. É proporcionar segurança com atendimento de qualidade e, principalmente, com muito respeito e atenção. Por isso, investimos em alta tecnologia e buscamos manter relacionamentos duradouros. Nosso compromisso é ser um parceiro confiável que compreenda as necessidades, a realidade e as condições de cada cliente para assim oferecer a melhor solução sempre. Entendemos os benefícios da pesquisa científica para a humanidade e isso nos estimula. Se hoje temos melhor qualidade de vida, maior longevidade, se vencemos um câncer ou fazemos uso de um remédio para dor de cabeça, é porque o avanço da pesquisa biomédica nos permite. Confiamos no trabalho dos pesquisadores, na ciência e na comunidade científica. E nos orgulhamos em fazer a nossa parte. Assim como você, somos apaixonados. Ciência é o que nos move, porque para a Lesco, pesquisa é para a vida.
Hello, everyone. We are back from the 1 hour break and welcome again. So, my name is Rafael, and I'll be the chair of this session together with my colleague, Dr. Renata Medeiros from FIOCRUZ.
Hi,
And,
everybody.
and before we start, I would like to remind everyone that the link for the certificates will be posted in the YouTube chat after this lecture, okay? So, it's a great pleasure for me to introduce our next speaker, Dr. Paulo Gavaia. Dr. Paulo Gavaia is a researcher at the Center of Marine Science Bioscale Lab and invited professor at the Department of Medicine and Biomedical Science, University of Algarve. With over 20 years of experience in the use of fish for research and development of industrial process, he is co-author of more than 80 scientific articles, coordinator or member of more than 20 scientific research and industrial cooperation projects, has won international patent and 11 industrial research contracts developed in the last 5 years. So thank you very much, Dr. Paula, and welcome to our webinar.
Hi, thank you so much for the presentation. It was very kind to receive this invitation, and it's a great pleasure to be able to share our work to such a great audience as you could gather with this webinar for Zebrafish. So I don't know if I can start. You let me know
Yes, please, you can
whenever I
start.
can start. Okay. So the title of my work is using zebrafish as a tool in bioprospection for osteogenic molecules. And I will talk a little bit of the work that we have developed in the last 5 years in the Bioscale lab, since we started to get interested in all the bioprospection and the interest in discovering molecules that can have, or extracts that can have a good effect, or meaning osteogenic effects on bone development. And how did this came out? So a little bit of introduction to our work is we have been working with bone biology and skeletal biology for many years now, and we started by using fish, several teleost fish, and we entered the world of zebrafish in around the year 2000. And by then we discovered the great advantages of working with this model, and we started to invest a lot on working with zebrafish, discovering new tools to work with zebrafish. And in the last since 2015-16, we started in collaboration with some project partners to use zebrafish in the bioprospection for molecules. So we had the opportunity to test the effect of molecules or extracts from natural origins that our partners were developing, and to test those to see if they would work both in vivo or in vitro, since we have developed cells derived from the skeleton of teleost fish. And from this, it began this story, and we found out that just by applying the drugs or the molecules or the extracts to the fish, it was more complicated than we thought at the beginning to have a good assessment of the osteogenic potential of these molecules. So, how did this came up? There are a lot of bone disorders nowadays, and there are available treatments. The most well-known, probably, and the one that affects most people is osteoporosis, that eventually everybody with aging gets to a point where they lose a lot of bone mineral density, and they get to a point where they develop osteopenia or osteoporosis. And nowadays there are several treatments or nutraceuticals that can be used to prevent osteoporosis. Namely, the ones that I have there, like vitamin D, bisphosphonates that are the most used drugs for the treatment of osteoporosis, RANKL inhibitors like the denosumab, estrogen receptor modulators, strontiuranolate that has given some problems in the recent years and that's been almost eliminated in many countries. or even the hormone calcitonin from salmon that is used as a nasal spray. The objective of using these drugs is to decrease bone turnover, especially to decrease osteoclast differentiation, reducing the breakdown of bone by the osteoclasts, or to improve osteogenesis, meaning to improve bone formation and to improve bone mineral density. However, there are a lot of undesirable side effects from the use of these drugs, like the osteonecrosis of the jaw that is very well described on the use of biphosphonates, gastrointestinal irritations, thrombotic events, diarrhea, nausea, some problems with the airways when you're using the nasal spray of calcitonin. So there is a real need for the development of novel molecules and molecules that promote bone formation with bone anabolic effects, and preferably molecules with a natural origin that are known to have less side effects than the drugs that are currently in the market. So we have entered this drug discovery on a phase that is on the beginning of the pipeline of drug discovery, on the pre-discovery, meaning on the screening for compounds, on the molecules that can have an effect for the desired target. Traditionally, this screening has been done in vitro using the cell systems, and you're using the in vivo animal models like rodents, sometimes using birds like the chicken. But we have seen that by using zebrafish, we can use much more specimens per screening, and we can do a faster screening. And I hope I would be able to show you this during this presentation. So in the last few years, and studying this problematic, we have seen that unlike the classical models, the use of zebrafish can be very advantageous, although it just started to be used recently. So in 2014, we released a review paper. It was edited by Vincent Lézé, one of our research colleagues here at FIOCRUZ, where we saw that fish is a very good model to study the bone diseases and to do this screening testing programs to discover new drugs with osteogenic or osteotoxic activities. And our colleague Ignacio Fernandez, that now has left our group, he was a postdoc here, now he got a position in Spain. He showed also that zebrafish is a fantastic model to assess the osteotoxicity of natural pollutants and of chemicals that occur in nature. And why is this so? Because although fish have diverged long time ago from the lineage of tetrapods, about 420 million years ago, all the mechanisms and the tissues that form the skeleton are pretty well conserved. So most of the skeletal tissues, the cell types, are pretty similar to what we see in land tetrapods and in humans. So, but why the use of zebrafish? This similarity to humans that I just mentioned, they are able to absorb the chemicals directly from the water, which is very simple to expose the fish. You just put whatever you want to expose in the water where the fish are swimming, and they will absorb it from the water. The larvae are, as you all know, almost translucent during the early development, so it's very easy to observe the developing organs. And in our case, particularly the skeleton that starts to form early, it's very easily observed during the early stages. And since fish were some of the first vertebrates to develop a bony skeleton, it will bring some evolutionary perspective into the mechanisms of vertebrate and tissue mineralization and how they are maintained throughout the evolution of all the vertebrates. And since it is small, it's a robust, it has a large progeny, it has a rapid development, it's not very difficult to maintain and not very expensive to maintain, which is also very important. It allows for us to develop medium to large scale screening programs for new molecules using very different sources of molecules or extracts, as I will show you later. So in the beginning, we had to understand how the skeleton develops in zebrafish, and it is pretty well characterized. We know that the skeleton starts to form very, very early. At around 96 hours post-fertilization, we already have the first bony elements forming, and we will take advantage of the formation of these first bony elements, like the operculum bone, that is a dermal bone that forms on the surface, and it is very easy to analyze on a developing larvae. And by about 19, 20 days, the zebrafish has already a fully developed skeleton. It's not fully mineralized yet, but all the skeletal elements are already present in the skeleton of zebrafish. It means that it has an advantage. We can see all the skeletal development in an organism that is developing externally, unlike mammalian models, and it is relatively fast to reach a fully developed skeleton. And the types of cells that maintain this skeleton are pretty similar. So in this image, we can see the types of cells, the four main types of cells that occur in bone. On one side, we have the osteoblastic lineage. So from mesenchymal osteoprogenitors, we have the development of pre-osteoblasts and osteoblasts, or the bone-forming cells, that are the ones that are going to deposit the matrix and form new bone. These osteoblasts will either lose some of their metabolic activity and become bone lining cells, or they will become buried in the matrix and become osteocytes, or the bone cells. On the other side, we have the cells that eat bone or degrade bone, the osteoclasts. These osteoclasts are formed with the monocyte lineage, so they have an hematopoietic origin, and they will be recruited by action of the osteocytes whenever they sense damage or any problem with the bone where they are inserted, or when there is apoptosis of these cells. Osteoclasts will come, will remove the bone that is damaged, and then osteoblasts will come again and form new bones. So all the bone health is dependent on a concerted action of both these lineages, bone-forming cells and bone-resorbing cells. And when we look at fish, what is the main differences that we can see? We know that some of the fish that have evolved and are known as the modern advanced teleosts have developed a type of bone that is acellular. It means that this bone lacks osteocytes. It is the case of some very well-known fish, as the gilt-head sea bream, all the sea breams, all the tuna fish, for instance. However, zebrafish belongs to the basal teleosts, and these basal teleosts have maintained the original feature of an osteocytic or a cellular bone, which means that they retain the osteocytes in their bone matrix. So this bone of the zebrafish is more similar to the bone that we observe in land tetrapods, including humans. So it's a good model to study all the skeleton, including all the cell types that you can find in humans. We have made a small table where we can compare. I'm not going to read all these, as you can imagine, but in this table, it is possible to see the similarities and some of the differences between fish skeleton and human skeleton. I would just like to emphasize the major differences. And the major differences is that in fish, the development of the vertebral column occurs in a different way. Instead of having an endochondral ossification of the vertebral column, meaning instead of having a vertebral column where the bones first form as cartilage and then this cartilage ossifies and becomes bone, in the fish, we have a perichordal. It means that is around the notochord, the embryonic notochord, that the ossification of the vertebral column will occur directly. And the types of cartilage. So in fish, we have described around 16 or more types of cartilage, while in mammals, we can find the description of 3 types of cartilage. But this is absolutely no problem when it comes to all the mechanisms and all the molecular mechanisms that regulate development of cartilage and bone. Okay? For studying the skeletal features of the zebrafish, here I have a table that was made by Eckhard Witten, colleague researcher, where he describes very well the types, some of the types of cartilage that we can find in fish. And this is very easily observable by histological section. And we can study the skeleton of zebrafish by using techniques like X-ray, like micro-CT, or directly, when we are talking about larval stages, by using the staining, the double staining of the skeleton using alizarin red to observe bone and alcyon blue to observe the cartilages. And this is one of the techniques that we'll be using the most during the systems and techniques that I will describe in this presentation. So we have, based on this fish, we have developed some systems that allow us to quantify the action of molecules on bone of the fish, depending if we are studying early stage or a late stage. So when we are studying an early stage, we will focus on the developing operculum, which is a bone that will form on the surface of the fish, and it's easy to observe. But if we want to study an adult, we can use skeletal deformities affecting the axial skeleton, the mineralization of these vertebrae in the axial skeleton. We can use the scales, and we can use the regenerating caudal fin of the zebrafish. And here we can see some images of some of these systems. So we can see the development of the head skeleton, the cranial skeleton, with calcified tissues in red and cartilages in blue. We can here very well see the opercular bone forming on the head of a 16-day-old larvae. We can see a regenerating caudal fin with already a large portion of the regenerated rays or lepidotrichia formed, and the contour of still non-mineralized tissue. We can see a scale that is possible to culture as an explant or to use as a regenerated scale in vivo in the fish, and we can use also fish cells that are derived from bone tissues, and they are able to calcify and will be able to apply all the treatments that we do in vivo also in vitro. And actually, we have already applied most of these systems to different strategies of testing. In this case, it was testing for toxicology, so the toxic effects that some chemicals and some organic pollutants can have on the development of bone. And you can see deformities in the skeleton. You can see malformations in the opercular bone. You can see increased regeneration or a demineralization of the scales. As you can see here, there is an increased activity of osteoclasts and alterations of the regeneration. And this means that the systems that we have developed, they can be used both for discovering molecules that will improve bone quality, but they can also be used for assessing the toxic effects of contaminants in nature. And actually, we have been using the same systems to do both things. But what do we have to do? So, by having molecules, extracts, drugs for repurposing, using libraries of molecules that are commercially available, we can apply these to fish, but then we have to understand what are the cellular and molecular players that are being affected, and we have been using transgenic zebrafish lines to understand how the osteoblastic cells are behaving when they are exposed to a given compound. We have been using reporter arrays like the signal that has several signaling pathways that are arrayed in a plate, and we can understand how these pathways are being affected. And by using techniques like gene expression, like mass sequencing, we can try to understand what are the pathways and the molecules that are involved in the response to these molecules. But we have to study using in vivo systems. And these in vivo systems will be using most of the life stages of the zebrafish. And what are we studying? We have been studying molecules mostly derived from natural sources, like derived from algae, derived from terrestrial plants, but also from microalgae and derived from marine invertebrates. Since we are working in a center of marine sciences, it's relatively easy to get collaborations to get this kind of plants. And before it has been described already, some possible mechanisms or some possible systems to study the effects of given compounds on the skeleton of zebrafish. The first work was made by Fleming in 2005, where he showed that by using the calcitriol, the active form of vitamin D3, and after 6 days of treatment, they could get a very good increase on the mineralization of the cranial structures of the zebrafish. And soon after, in 2008, dorsomorphine was used to 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 test a new system where it was proposed that the rate of development of the vertebra around the notochord could also be used for assessing the osteogenic effects. However, when we started to analyze these kind of systems, we understood that they were not very reliable. There was a huge inter-specimen variability. So we had the need to find a way to try to assess this in a more quantitative and less variable way. And for that, we started to develop systems. And now, at this moment, we have developed several systems on the early development, where we use the opercular bone. We can use the developing axial skeleton, and in adults, where we are using the caudal fin regeneration and the scales either as explants or as regenerates. And I've also been using gene expression either by using the normal molecular techniques or by using transgenic reporter lines. So I will talk a little bit about the first system and how we developed it. So the operculum, you can see it here in the surface of a fish, is a flat surface bone, is very easily identifiable by the shape on the surface of the bone, and is easy to monitor by using fluorescence microscopy. We stain the fish with alizarin red, and the alizarin red is a fluorescent dye that will allow us to observe the surface of the fish with very clearly. We place the fish laterally, and it's possible to observe and to measure the operculum. So this work was developed by Marco Tarasco, one of our PhD students that is currently finishing his PhD, and he did an excellent job in developing this system. And we thought that it would be a very, very high potential system to analyze. So what we did was to first to make a timeline of the skeletal development of the operculum. So we analyzed the development of the operculum until 15 days, and we saw that there could be some interspecimen variability of the area of the operculum. Whoever has worked with zebrafish larvae, they know that larvae have different rates of development. So we have to find an optimal time window where to analyze the opercular bone and reduce this variability. And then we had to choose what were the best parameters for morphometric analysis of this opercular bone, and use other measurements for making a normalization. And we took several measurements from the area of the eye, the length from the snout to the clatrum, the area of the head, the iris, etc. But what we found is that there was a very good correlation between the area of the head and the area of the operculum throughout development during the first days, okay, with a HAR of 0.94, which is very good. We could also use the length of the snout clatrum or the area of the eye, which is not very bad, but consistently the area of the head was the best measurement to normalize the development and the area of the operculum. And what we saw also was that during this early period of development of the operculum, meaning before 4 to 6 days of development, was where we had a higher increase in area of this structure. So we could fairly easily make an assessment of any effect that any molecule could have on the development of the operculum on this very short period. And in fact, it's what we have been doing until now. We have been exposing fish only until 6 days, with another advantage is that until 6 days, you don't really need to feed the fish because they are still feeding on the internal yolk reserves. So all the effects will not come from nutrition, but will come from whatever is in the medium of the fish development. So for this, we made several trials and we made several tests of exposure, increasing the exposure time or testing different onsets of exposure. And we really saw that the best correlation was done with exposure starting at 3 days post-fertilization and exposures during 3 days, meaning that we make just a very short exposure and the effect is quite reliable on our results. And you can see that here, this was just a test to see if the system was working. When we exposed the fish with the vitamin D, the calcitriol, we could get an improvement of about 50% on the mineralized area of the operculum just after 3 days of exposure. And we also needed to be sure that this effect was due to bone-forming cells. And for that, we did a time course of the activity of osteoblasts and the onset of osteoblastic cells on the fish. And what we saw is that osteoblastic cells do not appear before 13 days post-fertilization, which means that is much after the end of our experiments, meaning that all the effects that we will be seeing on the operculum are due to the effect of osteoblastic cells and not to osteoclastic cells. So we created a pipeline where we start with a primary approach that can be done with a high throughput screening. So we are testing a low complexity bone system, meaning we are testing just the growth of the operculum. It's an assay where we are going to evaluate the bone growth potential of this structure. As I told you, it will be mostly osteoblasts responsible for the development of the structure. It's a short treatment. We can use a relatively high number of animals per well. We do this in 6 well plates where we can put approximately 15 larvae on each well with a volume of, with a volume of 20 milliliters. And we always use vitamin D calcitriol as a positive control to assess if the effect that we are seeing is really due to a positive growth of the operculum. And we are always using the corrected opercular area, okay, by dividing the opercular area at area on the treatment divided by the control. So we have a difference towards the control. And this is how it is processed. This is the pipeline of all the process. So we get larvae at 3 days post-fertilization. They are still not feeding. They are almost stopped in the bottom of the tank. We array them in these 6 well plates and we expose to whatever molecule or extract that we want to use for treatment. And after 3 days of treatment, we will make live staining using alizarin red. We can expose the fish to the dye. All the calcified tissues will get stained in red, and then we can observe this red staining by using fluorescence microscopy. Then we make image acquisition and make analysis of these images. Okay, so we use a fluorescence microscope, and this is the kind of images that we get, and then we can measure fairly easily the area of the operculum. And this is the kind of results that we get. So in a control, we can see a normal operculum. In a pro-osteogenic molecule, we see an increase on the area of the operculum. And if we have a toxic or anti-osteogenic effect, we can see a reduction on the area of the opercle. As a proof of concept, we tested two molecules that are known to be either osteoactive or osteotoxic, and this is the result that we get, that we can see an increase, a dose-dependent increase on the operculum area with calcitriol, with vitamin D, and we can see a dose-dependent decrease on the operculum area by using cobalt chloride that is known to be osteotoxic. So this proves the suitability of this system to test molecules on this. And we can also use our transgenic lines that are markers of osteoblasts at different stages of maturation, like the SP7 and osteocalcin transgenics, where SP7 is marking early osteoblasts and osteocalcin will be marking mature osteoblasts. And we can use these transgenic reporter lines to investigate where the molecules are affecting osteoblasts, in what stage of development of the osteoblastic lineage the molecules will be acting. It can be earlier if they are affecting SP7, or it can be later if they are affecting osteocalcin. And we have crossed these two transgenics, so we have a double transgenic where we can see the labeling of both SP7 and osteocalcin positive cells. Okay, so these transgenic lines, they are very easy to maintain. They are exactly the same as any other zebrafish larvae, and they will respond in exactly the same way. But it allows us to investigate what effects these molecules are having on this specific lineage. Okay, so this is what we did. We have the two transgenics, we cross them, and after we make the exposure, we make the data acquisition very similar to what I showed you that is done on the wild type fish. But on this case, we will also acquire the areas or the number of fluorescent pixels that correspond to green if it is for osteocalcin, or to red if it is for Osterix sp7 positive cells. And we can see if there is a difference on the maturation of the osteoblastic lineage by then making a ratio between sp7 and osteocalcin positive cells. We can see here an example. By using calcitriol, we can see that there is a higher effect on osteocalcin than on Osterix. Okay, and it was proven by both by fluorescent area and by the number of pixels. We also tested this at different time stages, and we can see that by using an anti-osteogenic drug like the cobalt chloride that I showed you previously, it would also give us a similar result to what we saw. In this case, there is a huge decrease on the osteocalcin positive cells, as you can see by the decrease on green signal on this bone. And the other system that I will focus today is the caudal fin regeneration system. So this caudal fin regeneration system was developed by João Cardeira. João Cardeira was a PhD at our lab. Right now, he's a postdoc in Germany. And we have taken advantage of the very fast regenerative capacity of the caudal fin of zebrafish. The zebrafish can regenerate very fast. You can see here an image that is just 5 days post-amputation, and it has already regenerated a large part of the lost caudal fin, and it will respond to drugs, as you can see here. So, but what have we done? What was behind this? We first had to understand what mechanisms are involved in tissue and bone regeneration. Can it be used for drug discovery? And can it be the basis for novel treatments for degenerative or traumatic diseases that occur? So by studying these, we have applied molecules, different agents in the different conditions, and analyzed the effects on the performance of the regeneration of the caudal fin. Okay, then we analyze overall regeneration, and the outcome is de novo osteogenesis and mineralization. This is what we will really analyze. So First, what we have to do is to measure the mineral deposits. As you can see, this is also fluorescence images of alizarin red stained fish. We have to disregard the effects in ray thickness, and this is a pretty labor-intensive process. Okay, so we need to find ways that are more easy to analyze this. So we had a challenge: how many specimens we can use to have reliable results? Do they need to have identical agent size? What are the differences in size translating into different regenerated areas? So, meaning we need to make standardization and normalization of the results. Okay, but how? We need to have regenerated areas. We need to analyze the weight of the fish, the length of the fish, the stump width, to measure the several morphometric proportions like we did for the opercular previously, to try to understand how to normalize these intrinsic animal differences and to normalize then the regeneration process. We know that this process is quite fast. At 24 hours, we already see the regenerate forming at the tip of the ray fin. At 48 hours, mineralization starts. At 72 hours post-amputation, the segmentation is also starting of the bone. At 96 hours, we have bifurcation beginning, and by 2 weeks, we have a morphologically complete fin, meaning a fully regenerated fin. So we started by trying to understand what this regenerated area and the regenerated mineralized areas would tell us. So we measured the regenerated area, as you can see on the image in the left. We measured only the mineralized areas, meaning the area of the rays that were already remineralizing, and we also used the ray width on the pre-amputation plane to normalize then this mineralized area of the rays. And what we tried to understand what was the best correlation for normalization. So this is the unnormalized area. When we normalize it with the length, we have a fairly low R. But when we started to use other parameters like the weight or the stump width, we had an improvement on this. The best parameter actually was the weight of the fish. But we decided that it was probably not the best practical parameter, because when you're making an amputation experiment, you have to remove the fish from the water, we have to reduce stress, try not to influence too much. So we end up deciding that the stump width was the most high throughput, because it's fairly easy. We amputate the fish, we image the fish, and then we can make the measurement later. We don't need to be stressing the fish further and keeping the fish out of the water more time than it's really necessary. And by additional square rooting, we could improve a little bit these parameters, and it gives us a better correlation, so a better R for making this normalization. So actually, right now, what we are doing is using this parameter, the square root of the regeneration area divided by the stump length, meaning the length at the amputation plane. And we can use then these results to understand how does the overall regeneration is working, how de novo mineralization is progressing along the regeneration, depending on where the position of the points of each fish in each individual fish will be. We will know if we have over mineralization or under mineralization. And then we can also use live specimens, okay? Because we can make a live imaging to these amputated fish, let them regenerate, make them just make imaging of anesthetized fish, and follow up the same specimens, meaning that we can reduce the number of specimens to be used in one experiment. And here you can see some proof of concept using molecules with mineralogenic action. We can see that retinoic acid provokes a huge fusion effect on the developing rays. after 5 days of regeneration, or 120 hours, you can see that there is a huge decrease on the formation of the rays, and the rays have a morphogenic effect, so they are all fused and don't grow in length. So we have used this for studying timings of regeneration, mispatterning of the regenerating lipidotrichia, the caudal fin rays, and also the number of segments and the length of the segments can give us an idea of how this process is being affected when we expose the regenerating caudal fin to, a given molecule. So the position of the bifurcation and the number of segments until the beginning of the bifurcation give us an idea of how the patterning of the de novo forming rays is occurring. Okay, and also the mineral organization, as we can see on the retinoic acid example that I showed you, or by the reduction of this mineralization here by exposing this to a bisphosphonate for 5 days. And to simplify all the analysis of this data, Marco Tarasco has developed a semi-automated method using ImageJ, where we created a group of macros for simplifying the analysis process, and they called it the ZFBone Toolbox, okay, which is a tool set that can be downloaded. It's free on GitHub, or it can be used as a supplement for ImageJ, and it will simplify and automatize the analysis process. It can be used for the operculum, for the regenerating caudal fin, but also for scales, for ray bone intensity. So it's very easy to use. It has a help button that will explain very well how to use all the features of these macros, and it will simplify and speed up the process. And finally, I will talk very briefly about the axial skeleton. So the kind of exposure that we do to study the effects on the axial skeleton is very similar to what we did for the larvae for the operculum. So we expose larvae from 3 days post-fertilization. We allow them to grow until 30 days post-fertilization, where we have a fully formed individual. It's a juvenile with a fully formed skeleton. And then we will double stain the skeleton to observe deformity levels by just placing them on a stereo microscope. And here we have some examples of these deformities that are developed on zebrafish. Here we can see vertebral fusions and arches fusions. Here you can see arches that are not forming correctly on the caudal fin area and the caudal fin vertebra that are fusing and deforming all these rays, or here some opercular malformations. And a more practical example is from a thesis of a student that just finished her BSc thesis, where she exposed larvae to an organic pollutant, and with increasing amounts of this organic pollutant in the water, we could see an increase on the number of deformed individuals. And here we can see some examples, also the gravity, so the severity of these malformations that we could observe on the fish were also increased by increasing the amount of this pollutant in the water. So let's now see some applications that we have done to these systems that I just showed you. So I will mostly focus on the osteogenic activity of natural compounds. So we have made some work in collaboration with colleagues from a project that just finished 2 years ago, and we were studying the effect of extracts from green macroalgae. We could see that extracts from Codium fragile and Codium rupestris had a fairly good pro-osteogenic effect when they were increased. Okay, so you can see a dose-dependent increase until 5 micrograms per ml, and then some decrease that probably might indicate that there is some toxic effect on this extract. Also, on Codium rupestris, on different concentrations, you could see a fairly good increase. You can see that it's over 50% increase on the opercular area towards the control. And you can see that is even higher than our positive control, the vitamin D, in both cases. Also with red macroalgae, this was a work done in collaboration with Queen's University in Belfast, where we had access to some extracts from algae collected both from the coast of Ireland and some red algae collected in a project where they have a collaboration also. So we had studied the pro-osteogenic effect, and we could see that both Ceramio secundatum and Plocamium had a positive effect on the bone development, here by analysis of the operculum, and also Ceramium pallidum from the Irish coast had a pro-osteogenic effect. So these are all extracts that I believe our colleagues from Ireland are now further characterizing to try to understand what are the molecules that are responsible for this pro-osteogenic effect. We have also studied the effect of environmental pollution. Okay, so in this case, the effects of methylcolantran, the osteotoxic effects of methylcolantran in fish, and the anti-osteogenic activity of cadmium contamination using this system of the operculum. And you can very easily see here, it's an inverted graphic. So the highest concentration is the first, and you can see a very high reduction on the area of the operculum towards the negative control. I'm not even talking about the positive control, okay, in both cases, both in cadmium and in methylcolantrin. We also have tested together with some colleagues from Portugal in Alentejo some extracts from cardamom and eucalyptus, which is some very well-known plants, and we could see positive effects on the activity on growth on the extracellular matrix using cardamom extracts and using eucalyptus extracts, but stronger in cardus than in eucalyptus. These were dichloromethan fractions of these extracts, and we were quite happy with the results. So there was over 300% increase on the osteogenic activity. Probably the tripernic acids, eucalyptus, and not cyanopicrine in cardon, because we can see the cyanopicrine had enhanced the osteogenic effect. It was exactly the opposite of what we wanted to see. And we tested then some molecules that could be responsible, and we saw that ursolic acid, which is a molecule that is abundant in eucalyptus and cardamom, can be one of the responsible for this pro-osteogenic effect that was observed. So another of our colleagues, Joana Rosa, she's a researcher still in the Bioskel lab. She has been testing different extracts from different origins. In this case, she have tested extracts from bacteria, 12 marine and freshwater strains where acetonic extracts were prepared, and they were tested all at the same concentration of 10 micrograms per ml. And we could see that 3 of the planktomycet strains tested and 1 of the proteobacteria strains showed an effect over the positive control. So the positive control is this dotted line that you can see here, and if the pro-osteogenic effect on these strains were over the positive control, which indicates that they can be good candidates for developing or for trying to discover what are the molecules they are being responsible for these effects. So this extracts was selected for a secondary testing, secondary screening. This secondary screening was done using adult fish to see if the effect was maintained from larvae to adults using the cow dolphin regeneration. So it was also proven that 1 of the planktomycet strains still showed mineralogenic activity, pro-mineralogenic activity, this B2 strain, on the cow dolphin, meaning that this is probably the most promising strain to go forward with this work. She has also been testing extracts from cyanobacteria, 8 strains with 9 to 11 fractions with different polarities, all tested at the same concentration. And also she could see that 3 of these strains showed anti-osteogenic activity, meaning they decrease the area of the operculum. You can see them in light blue. And 4 of the strains had a pro-osteogenic activity. You can see them in the dark blue bars. Also, she has been testing different species of microalgae extracts. So from these microalgae, 3 different extracts were prepared from each of the species, meaning that there was 36 extracts with water, ethanol, and ethyl acetate as solvents, and 3 concentrations of each of these extracts was tested. And we could see that some of these microalgae had fairly good effect, and it was mostly in the ethanolic extracts, although some water extracts also showed moderate pro-osteogenic. The stronger pro-osteogenic effects were always in the ethanolic extracts. Okay, in total, 17 of these extracts showed pro-osteogenic activity, while only 2 were proven to be anti-osteogenic. So 6 extracts were selected for going further for a secondary screening, using the extract and the calcitriol as a positive control. So again, using adults and the caudal fin regeneration, and 5 out of the 6 extracts that were selected showed to be mineralogenic. So they maintain the pro-osteogenic activity and the mineralogenic activity from larval to adult stages. Also, another student of ours, Alessio Carletti, it's a PhD student on a Marie Curie project. He has been doing the study of some extracts from marine invertebrates like holoturians and tunicates. This is a work together with the Portuguese Institute of the Sea and Atmosphere in Lisbon, where they have been collecting these specimens and they have been testing activities from extracts of these specimens, and we decided to test it also on the osteogenic activity. And what we saw is that some of these extracts, again, mostly the ethanolic extracts, meaning the gray bars on this graph, showed some pro-osteogenic activity in 3 of the tunicates and in 3 of the holoturians tested. This, together with other activities like the strong antioxidant capacity, the polyphenolic contents, and the anti-inflammatory effects that these extracts have shown, they make them very good candidates for further studying. We just published this testing, one of the microalgae from a group here at the CCMR, the Marine Biotech, and he has been testing the ethanolic extracts of the CTP4 strain. And we have seen that it is a very promising strain since it shows very good pro-osteogenic effects on the skeleton. You can see here the comparison between a larvae, the operculum of a larvae control, and the operculum of a larvae treated with 200 micrograms per ml of this extract. There is a huge increase on the size of the operculum. So we have been trying to understand what are the cells, the point of the cell cycle of the osteoblasts that is being affected. There was some difference on the osteocalcin cells, but it's mostly at the early differentiation that these extracts are having an effect. Okay, so meaning it's the sp7 transgenic that is giving the higher increase in signal. So we have chosen to fractionate this extract by HPLC. We have tested the 15 fractions that we obtained. We retested them using the operculum system, and we chose 2 promising fractions, since they had no toxic effects. Although some of these fractions had a very high effect, they also showed toxic or teratogenic effects. So it shows some that have a good effect but no adverse effects to go forward. We have fractionated them further by LCMS, and we have identified a group of compounds by the similarity match and that were not present on the neighboring fractions. So they were unique to the fractions that we were studying. And we have selected right now a list of candidate compounds that were reduced to 15 compounds that are being tested right now. And we are trying to understand if any of those candidate compounds is the responsible for the effects that we are seeing on these extracts. So this is the end. I would just like to thank all my colleagues that have done the work and have participated in the studies of this presentation that I showed you in this presentation. Of course, to thank all the funding bodies and the projects that have collaborated with us and people that have provided us with the extracts that we have been using, or the organisms that we have been using. And to remind you all that we are organizing a course where you can learn all these techniques for screening osteogenic compounds using zebrafish. We will be holding a course from the 8th to the 10th of September this year. The deadline for inscription is tomorrow, so if anybody is interested, hurry up. You have the information there. And thank you all for listening to me.
Okay, thank you very much, Paulo, for very interesting and inspiring talk. I really enjoyed how you develop your pipelines, and the applications was very, very nice. And we have a couple of questions.
Okay.
So the first question is from Veronica Lombardo, Veronica Lombardo from Argentina. Great lecture. What zebrafish line is used for alizarin red S staining?
We are using...
there are more. Could be used wild type line. Is necessary to avoid autofluorescence, or is needed to use Casper mutant or another one?
No, we are simply using the AB wild type. So it's one of the most common wild type lines used all over the world. Okay, the AB line can be easily purchased from any repository in ZFIN, in the EZRC. You can find the AB line. Okay, it's a pretty simple wild type. There is absolutely no need to remove pigment. For the operculum, at 6 days, there is absolutely no interference. There is very little autofluorescence using the red channel. Although we are making live staining of the whole fish, you can observe some background, but since it is a very superficial bone, the focal plane allows you to perfectly identify the structure, to measure the structures without any problem. The caudal fin is a very thin structure, so there is absolutely no need to remove any pigments at all. Okay, just use the fluorescence imaging, and all the pigments will be invisible under fluorescence.
Good.
I hope I answered the question.
So the next question is from Bruno. It's a very nice question. He said, hi, Dr. Gavaya, great talk. Do your group already seen if these transgenic zebrafish with larger operculum can have a higher ventilatory frequency and consequently cope better with some stressors?
Actually, no, we don't. We are not using any physiological approach to this. And actually, all these fish, when they developed eye or operculum, means what it means for us is that all the skeleton is developing at a higher rate. So we'll have a better development of the skeleton. We have an increase on the activity specifically of the bone cells. But sorry, Bruno, we have not been following any approach to understand if it will influence or not the ventilatory frequency. Actually, we have been testing in some way the capacity of fish when they are exposed to molecules to have differences in locomotory behavior. Okay, but it's only locomotory behavior. We have a Xantix apparatus that allow us to follow the trajectory of the fish, but we have not correlated it with a higher increase in the opercular bone, because normally when they have a higher opercular bone development, if we are going to analyze the rest of the skeleton, we also see there is an increase of the rest of the skeleton, meaning that the whole mineralization of the skeleton is occurring at a higher rate, and that's what we are really looking for.
Okay, good. Thank you. We have a question again from Veronica from Argentina. And she said, how is evaluated the antioxidant activities from marine green microalgae on zebrafish?
Okay. Basically, we are using commercial kits for understanding the antioxidant activity. So you get the larvae, we make a extract. Okay, we have to macerate the larvae, and then we see how is the antioxidant state. We are using several kits, okay, from the COX activity to the superoxide dismutase, and okay, but all commercial kits. Very, very simple to use. It's not a secret technique.
Okay. So next question is from Pierre-Olivier Angan. He said, great talk, Paulo. Very inspiring. Is there a rationale to search for molecules having an osteogenic effect from plants' algae? Previous findings or compounds from pharmas?
Well, pharmas that I know of, probably they have some pipelines, but they keep it secret. Okay, that I know of, they don't have any developed molecules derived from this algae or microalgae. We have started testing this. It was a little bit by chance. Okay, I have to say that, because this arose from the collaborations that we have been having, in some from European projects, some from a project that is the MED, or the Atlantic Space Area, where we have several partners from different countries. As you could understand by the talk, we have collaborations with many different places, and these are the people that have been providing the extracts. I can tell you that at this moment we are testing, for instance, fungal extracts from a group of collaborators in Italy that heard of our testing these screening systems and decided to try with us. So we started with the macroalgae and the microalgae exactly because our partners were studying other activities. They saw that there was the potential of having other activities, and we also were included in this testing pipeline. So people have been testing activities for anti-obesity, for dermatological problems, for diabetes. Okay, so many different purposes that have been testing these extracts. And since the extracts were available, why not including them on the pipeline and testing them also for osteogenic activity? Okay, many extracts that we test, and sometimes we receive groups of 200, 300 extracts that have been prepared. In all these, probably 3, 4 can have a moderate effect or no effect. We never know. And sometimes some of the extracts that we get, they have exactly the opposite effect. They have a completely anti-osteogenic effect, which is also a good result. Okay, don't get me wrong when I say that is osteogenic effect is always an increase in bone formation. Sometimes understanding what is there making a decrease on the osteoblastic activity is also good. There are some diseases like osteopetrosis, for instance, where you want to decrease bone density. It's not always a problem of increasing bone mineral density. I think I have diverged a little bit from the original question that was made, but I hope that in between I have answered the question.
Okay, Paulo. Beatriz Patricio said, thank you for the inspiring talk. I would like to know if there is zebrafish model of bone disease as osteoporosis or bone cancer.
Yes.
Do you have any work with this kind of models? Very nice question.
Yes. There are already many zebrafish models of bone disease, including osteoporosis. Okay, the first osteoporosis model was not created in zebrafish; it was created in Medaka. Actually, we have that Medaka model also in our lab. We have been using it to test. But you have both types of models. You have induced models. For instance, you can just treat the fish with compounds that are known to reduce or to induce osteoporosis, like glucocorticoids. Okay, just by treating the wild type fish with a glucocorticoid, you will induce an osteoporotic state, and that can be used as an osteoporotic model. But you have also models now that have an overexpression of transcription factors like RANKL. RANKL will increase the osteoclastic differentiation, so it will increase the resorption of bone, and it will promote a state very similar to osteoporosis. But you have models for osteogenesis imperfecta with the collagen 1 mutants. Nowadays, there are a very big array. And actually, we have just published a review article on that. I will be happy to send you that.
Great. Okay, then there's one question from Ragan Bukemaya. Could these osteogenic compounds be a more affordable solution to treat osteogenic disorders?
I really think so. And when I say I really think so, is because if there is availability, and this is also one thing when we are working with these marine derived extracts or natural derived extracts. If there is availability of biomass, like for instance the microalgae, this last example that I showed you, that is relatively easy to grow. If it is a biomass that can be propagated in high quantities, and if it is a simple extraction process, it can be used directly as an extract if the extract doesn't provoke any secondary effect on humans. It can be used not as a drug, but it can be used as a nutraceutical, okay, as a food supplement, which is fairly easy to prepare. It's much easier to have it on the market than a drug that has to go through all the pipeline of drug approval that can be extremely expensive, and us as a research group will never be able to enter on the further steps of the pipeline, especially due to the costs and to the long-term investment that you need to have. We would always need to have partnerships with the pharma industry to support all these costs. Okay, that's why we are placing ourselves at the beginning of the pipeline, but also because we are trying to find molecules or extracts that are possible to be either isolated or that can be synthesized. Okay, and if they are synthesized from a natural product, or they can be easily isolated from a natural product, I think they can be cheaper than what is currently in the market. Although we are not a pharma industry, of course, so I would not be able to tell you what are the final cost of all the development of one of these molecules until it gets to a drug. But it is in the numbers of several million euros or dollars to get to that point.
Okay.
Okay.
Paulo, we have maybe one more question. Giovanna Menezes. Congratulations. Opercular has been used as a parameter to evaluate disease-associated aging patterns in adult zebrafish. Is a question.
Actually, we haven't been using the operculum in adult zebrafish. We have been mostly using the opercle, the analysis of the opercle, to study during the larval stages. Okay, because in the larval stage, this is one of the first bones that forms at the surface of the fish. Is very easy to observe. When we are studying an adult, we observe the deformities of the operculum. And that's relatively easy because it's still a surface bone, and it's very easy to understand on the cranial structure of the fish what happened to the operculum. But not as a aging associated disease. Okay, actually, in our lab we don't work with not until now. We don't. I'm not saying that in the future we'll not do it, but until now we are not working with aging associated diseases, except osteoporosis, which is a consequence of aging, of course. But the operculum is not affected very much by osteoporosis. It's a dermal bone, not very exposed to osteoporosis. In fish, what will be mostly affected by this aging osteoporotic will be the vertebral column and the scales, okay, which will be the structures that will respond more.
Okay, we came out one last question from Hagen again. Have you tested the effect of these osteogenic compounds in the other connective tissues? It's from Chile.
From Chile. Well, only by molecular approaches, okay? Just by testing the effects on other connective tissues in gene expression pattern. Okay, so for instance, the cartilages are also affected sometimes, but since we are only on this screening pipeline that we are following, we are only visualizing bony tissues, because the staining that we do, the alizarin red, is fairly specific for calcified tissue. We are only analyzing the bones. But of course, when we try to understand further, we are also analyzing, when we make prepare RNA, and then we are going to analyze also markers for cartilage and for fibroblasts. So on that aspect, yes, we are analyzing the response of other connective tissues.
Okay, I think this was the last one, Paulo.
Okay.
Thank you very much again for your wonderful presentation. It was very nice to have you here.
It was a pleasure.
And also, if the students want to subscribe or in your course, I think it's a very great opportunity.
I think it is. It's already on the 5th edition. We have had a very good feedback from the people that were here. It's a 3-day course with a very strong practical part where people are really going to make all these steps that I showed here during the presentation. Okay, and they are followed in small groups by the people in the lab that are using this on a daily basis. So I think if people are interested, it's an excellent opportunity to come and visit us at the CCMR and learn some very interesting techniques.
Okay, thank you very much. Yeah, thank you.
Thank you very much, Dr. Paulo.
It was a very big pleasure, and I would like to thank you for inviting me for giving this lecture. It was a pleasure.
Well, our pleasure. Thank you very much, Dr. Paulo. Okay, people, we are done with the webinar for today. I hope you all enjoyed the lectures. I want to congratulate all the speakers for the excellent and very interesting subject we had here. I also congratulate the audience for being here. Stay tuned with us, and I hope you're enjoying. So just to remind you, we have posted the link for the certificates. So please fill in and tell us your feedback about the webinar, and also to get you get the certificate of participation. Thank you very much, and see you tomorrow. Bye bye.
Bye bye.
Trabalhamos diariamente para criar soluções e equipamentos para a comunidade científica, contribuindo com o avanço da pesquisa biomédica latino-americana. Nossa missão é maior do que somente fornecer equipamentos. É proporcionar segurança com atendimento de qualidade e, principalmente, com muito respeito e atenção. Por isso, investimos em alta tecnologia e buscamos manter relacionamentos duradouros. Nosso compromisso é ser um parceiro confiável que compreenda as necessidades, a realidade e as condições de cada cliente para, assim, oferecer a melhor solução sempre. Entendemos os benefícios da pesquisa científica para a humanidade e isso nos estimula. Se hoje temos melhor qualidade de vida, maior longevidade, se vencemos um câncer ou fazemos uso de um remédio para dor de cabeça, é porque o avanço da pesquisa biomédica nos permite. Confiamos no trabalho dos pesquisadores, na ciência e na comunidade científica. E nos orgulhamos em fazer a nossa parte. Assim como você, somos apaixonados. Ciência é o que nos move, porque para a Alesco, pesquisa é para a vida.